All right, so in the last video clip, we learned about some of the variation among the different types of neurotransmitters that are produced by neurons, and kind of based on some of their chemical classification. And here I'm just going to show you that different neurotransmitters can have different functions. So neurotransmitters can also be classified according to their function in addition to their chemical structures, what the molecules themselves actually look like. And so this is kind of linking back to video lecture 7, where we talked about how a presynaptic neuron may try to excite or stimulate a postsynaptic neuron, or in other cases, the presynaptic neuron may try to inhibit the postsynaptic neuron. And remember, when we say stimulate, it means the presynaptic neuron is trying to get the postsynaptic one to generate an action potential. It's trying to make that happen more easily. And so stimulation occurs because your postsynaptic neuron is undergoing depolarization. At least in that little local area, the membrane is starting to depolarize. Remember, though, you've got to get where, in order for an action potential to happen, that depolarization has to be strong enough, and it has to wind up diffusing, making its way all the way over to the axon hillock in order for an action potential to take place. And then when a presynaptic neuron is trying to inhibit a postsynaptic neuron, it does that by triggering hyperpolarization. So the membrane of the postsynaptic neuron becomes even more negative. Hyperpolarization means you're falling farther and farther and farther away from threshold and making it less likely for an action potential to take place. Now, interestingly, the same neurotransmitters, in some cases, the same molecules can be excitatory on some postsynaptic neurons, whereas on others they may be inhibitory. Whether or not a neurotransmitter is excitatory or inhibitory, it's all based on the receptor, across this. If you got an axon terminal here, and then here's the membrane of your postsynaptic neuron. This is our presynaptic over here. It's all about these receptors you've got over here for the neurotransmitter that are sitting on the postsynaptic neuron. So the neurotransmitters come out of the axon terminals, they diffuse across, and they bind to those receptors. So it's all about these guys and what happens after the neurotransmitter binds to those that determines whether or not the postsynaptic neuron membrane is going to depolarize or hyperpolarize. All right, and just as some examples, the neurotransmitters GABA and glycine are pretty commonly used in the central nervous system, and they're usually inhibitory, so they usually trigger hyperpolarization. On the flip side, there's a neurotransmitter glutamate, which is also an amino acid, and it's usually excitatory. Acetylcholine, which we learned about back in Unit 4, is excitatory at neuromuscular junctions. So in that case, your skeletal muscle fiber is the postsynaptic cell, and of course that is trying to trigger a muscle fiber to contract. But then on the flip side, over in your cardiac muscle, and in other parts of the body as well, which we'll talk a little bit more about later, acetylcholine is inhibitory. It actually makes it less likely that cardiac muscle fibers are going to contract, or contract as strongly as they normally would. So that's kind of interesting. Different parts of the body, the same chemical molecule can have different functions, and it's all about the receptor that it attaches to and what happens when that neurotransmitter binds to a receptor. All right, some neurotransmitters work through direct action. All right, and so that means the neurotransmitter binds to a receptor that is also a channel, an ion channel. All right, so the receptor is also an ion channel, like what we learned about back in Unit 4. So like they're showing you down here. Here's your receptor. The little yellow spheres there are supposed to be some the green spheres are a neurotransmitter. So when the neurotransmitter, the little green spheres, bind to the receptor, it causes the channel to open up, and ions are now able to move from one side of the membrane to the other. They're just kind of showing you a generic example in that particular case. And so since these are ions, that changes the voltage, changes the charge across the membrane, and so you can pretty rapidly have membrane changes. You can have rapid responses. These can be excitatory or inhibitory. It depends on how the ions are moving across the membrane. You know, what is the channel letting across and in which direction. So that's how acetylcholine works. That's also how the amino acid types of neurotransmitters work. Okay, and then some other types of neurotransmitters work indirectly, and I'm not going to go into details of this. Your textbook, if you want to take a look at some of the information in your text, and there are a figure in your textbook that shows this in a little bit more detail. But indirect action means, all right, so if we got a cell membrane here and you've got a receptor here, neurotransmitter binds to its receptor. The receptor is not an ion channel. What happens when the neurotransmitter binds to its receptor, this receptor now acts on another molecule, which may act on another molecule that could be inside the cell, and it kind of sets off a cascade where one step leads to the next and that leads to the next. You'll learn a little bit more about that when you take Biology 202 and you study the endocrine system, when you start looking at hormone type functions. The effect inside the cell can become amplified, which means maybe a little bit of neurotransmitter binding at the surface can actually trigger longer lasting effects, a longer response inside the cell. So unlike when acetylcholine binds a skeletal muscle fiber and it'll generate one action potential on the skeletal muscle fiber, and then if you want to keep doing action potentials, you need more acetylcholine to keep coming from that presynaptic neuron. In the case of these, you might have a low amount of neurotransmitter that binds to the postsynaptic neuron, but that might trigger some sort of longer lasting effect inside the postsynaptic neuron. It might change its behavior for a longer period of time. The ones that act like these are your biogenic amines, the neuropeptides we talked about on the last lecture, and then some of those dissolved gases like nitric oxide and carbon monoxide as well. So like I said, if you're interested in, if you like molecule type stuff and you want to take a closer look at how longer lasting effects can occur, check that out in your textbook in the section on neurotransmitters. All right, so again, please be sure to check the study guide for Unit 5, and I'll have some specific assignments for you about what you need to know for the exam on neurotransmitters. Next video lecture, we're actually going to get into the brain, which, as you might imagine, the brain is a pretty complex organ, and so we're going to go over some of the highlights of different parts of the brain and the functions of different parts of the brain, which can be pretty interesting. So we'll pick up with that info on the next video lecture.